So welcome everyone who's here, everyone who's virtually to the HCA general meeting. It's really fun to see everybody. Usually I get to split this talk with Sarah. I'm going to present alone because we wanted to make sure that this works smoothly, but she'll be here for the Q&A and throughout the meeting in many ways. And we also wish her, healthy, much health and a very fast recovery. Okay. So in this opening talk, I'm going to give you a very brief update on what's up at HCA, but I'm going to especially focus on how during the meeting we're going to be discussing these big questions that are looming for us right now as we're grappling with how we pursue the mission of the HCA, which is to create comprehensive reference maps of the types and properties of all human cells as a basis for understanding, diagnosing, monitoring, and treating in health and disease. The HCA community has grown and it continues growing. This was a pretty dramatic process over the last several years since we launched the Atlas collection. And what I think we're most proud of right now is that now it's a truly global footprint, and that includes the global south. So we now have leadership and a really vibrant and growing membership across the globe, including in South America, in Africa, and in the Middle East, 3 areas where we didn't have as many members when we just launched the HCA. And together with the members that we have in North America and in Europe and in Australia and in Asia, this means that when we build an atlas, it's diverse, both in terms of the scientists and in terms of the data, and that people are really working in the communities that we aim to serve. Overall, we just passed the 3,000 member mark. It's a real number because it's not 3,000, it's 3,009, and from 95 countries and over 1,500 institutes. Now, there's many ways in which we're organized, but one of the main ones is that people are organized in these 18 biological networks that Gary described to you as well. And that's really where the data gets collected. Each of these networks is generally focused on an organ or a system, but we also have a couple of networks that are devoted to overarching goals. For example, we have a network for genetic diversity to make sure that we're creating an atlas that's diverse genetically and ancestrally. We also have a network that's focused on human development, which is something that happens across the entire body and you can't really parse just by organs and systems. Across all of these networks, there's constantly data growth in terms of the number of individual samples, the cells, the numbers of samples from each individual. And I'm just going to pick on a few highlights because it's impossible to go through all of them. So for example, we've seen a ton of progress around the nervous system and in particular the brain. And this is really due to the fact that there is a major initiative funded by the NIH called the Brain Initiative. And Edlin, who's somewhere here, I saw him this morning, is going to talk this morning, a little bit more about what's happening there. And in particular, how things are even picking up more speed right now as the BICAN initiative has been started in order to build the ultimate human brain cell atlas. Another major effort is in the lung. This is also our first released integrated atlas. I'm going to talk a little bit more about this soon. But there's a few others that really stick out in big numbers. One of the most prominent ones is the immune one. Partly, this is because peripheral blood mononuclear cells, or PBMCs, as well as whole blood, are kind of easy to get from people. So you get a large number of those samples. But the second part of this is because immune cells are everywhere. So pretty much in every network, when we collect data, immune cells come along for the ride, and honestly, the tissue wouldn't have survived without them. So that's, again, a very large effort. This actually segs into also the effort to collect a genetically and ancestrally and geographically diverse atlas, which is also rapidly growing. Many of the first efforts on genetic diversity are focused on the immune system, and Chloe and Shyam, in some tag team effort, I'm not sure how we'll do it technically today, given that Shyam is one of our stranded participants, are going to talk to you a little bit about that this morning. It's also actually really exciting to see how rapidly other efforts have been progressing. So, for example, the oral and craniofacial network and the adipose network, they weren't as prominent or as active a few years ago when some of these other efforts have been going, but both of them have been progressing really fast. Or if you look at the reproduction network, some of the organs and systems that the team there is focused on are things that are actually very understudied in biology and that lead to a lot of unmet need. For example, endometriosis in females is related, of course, to our reproductive system. And so across the board, really exciting. And this is stopped moving. Let's see why. Hopefully now it moves, yeah. And not only are the networks hard at work at collecting data, they're also trying to build these frameworks, which we call roadmaps, to guide the work ahead. So 4 of these roadmaps have been completed and published for lung, gut, oral and craniofacial, and for development. More are forthcoming, and all of these are going to be reflected in the overall white paper that we're in the process of revising. It's also a great opportunity for me to just thank the coordinators of each of the networks. They work tirelessly and they work enthusiastically, and it's a lot of work to keep this going, and so we're very grateful for it. Okay, so now with all of these data growing rapidly, we of course have pivoted in the past year into what we call Atlas integration, which is also the first theme of our general meeting. Gary highlighted it for you as well. And in this morning's session in particular, we're going to be focused on what integration actually looks like. What does it look like now, and what do we want it to look like in the future, and what are the challenges between the now and that future? And so, as you may recall, as you may recall, in the previous general meeting, we kicked off a multi-group team to start on this effort. The first step was to pilot what we call the minimal viable product, or an MVP, of integration, and we chose 6 systems in what we called wave 1, the immune system, kidney, eye, gut, the nervous system, and the lung. So when we chose these, we had several different kinds of criteria. So for example, we wanted to have available and mature enough data to do an integration. We needed a committed set of bionetwork coordinators that were willing to partner in this effort. And also, we needed these to reflect somehow different kinds of organs or systems, and so different kinds of integration challenges. So we're not just repeating the same thing again and again and again. And Malte Luken is going to give us an update on this initial effort. Now, some of the integrations actually already have kind of their MVP pretty much there. I'm highlighting some images from the retina and from the lung. Others are ongoing, but everything is progressing really nicely. I promised you a little bit more about the lung because this was actually a first great example for us of what integration could look like, and also inspired us in feeling that we can really set to work on this across many different systems. And so the first integrated human lung cell atlas has just been published about a week ago, I think. Yes, Fabian, 2 weeks ago. About 2 weeks ago. And this atlas has a harmonized cell annotation ontology. It has harmonized data, metadata. It has diverse anatomic locations in an initial common coordinate framework for the lung. So it's a good substrate. And then the subset of the data served to build the core integration, and then it was extended with additional data sets using transfer learning, so that there's a total of, 2.5 million cells roughly, 50 different data sets, almost 500 individuals. These are pretty robust numbers. And then the team showed also how you would use an integrated atlas like this. You can query it in all sorts of ways. You can generate detailed cell annotations and markers. You can look at the effect of different parameters like age or BMI or gender on cellular profiles. You can relate GWAS genes to cells. You can discover new cell types. You can look for transitional states. Not everything is a cell type. And so this and many, many more, go read the paper, but also a nice template for us as we think about additional challenges in other networks. And so kudos to many, many people, Lisa, Malte, Fabian, Pascal, Jay, Sasha, Martijn as coordinators, but also the whole network and the many, many contributing labs. Now, there's a lot of work that remains to be done. First, to finish the first wave MVPs, and then to move them from minimal to maximal. But at the same time, we're kicking off 6 additional integrations: the heart, the liver, the skin, the pancreas, oral and craniofacial networks. These are all kind of similar. It's an organ or a system, but also the genetic diversity network, which does pose quite distinct challenges and is a very exciting move for HCA. Now, underlying any integration are 2 things. There's scientists and their human expertise, and there's computational tools and algorithms. And there's a large space of tools right now that are emerging in our community to do that. These are just a few illustrative examples that I think people are using quite heavily, and those include tools to generate references, to query them, to annotate cells, and to expand them as additional data is added. And regardless of the computational tool that you use, we need to put the knowledge that we have in the end into the right type of software platform. And so as the integrated atlases are getting built, our cell annotation platform, or CAP, led by Chloe Villani, who will speak later this morning, helps investigators create a reference for the cell identities based on molecular signatures. And instead of just making this a static place where somebody puts in the annotations, this is a living, breathing kind of platform, so that the platform is centralized, but it's really driven by the community that creates and explores and stores and serves these annotations at multiple levels of granularity, so that different people can provide different annotations. You can collect all of the information, and over time you can also train algorithms to do some of this work. And so it's really exciting to describe progress, but it's also important to remember that the key most word in a minimal viable product is that it's really minimal. And that means that there's a lot of difficult things ahead of us, and we should absolutely not be satisfied with what we have in hand right now. So I wanted to highlight some of the open questions that we feel we really need to tackle in this coming year or couple of years as we build this first Atlas version. And one that is top of mind right now, and I already alluded to, is the concept of defining integration between the clean and simple, strict definition of cell types. So saying all of these cells belong to a category and they have a name is an important thing. We use it, it's important for us. Things that have a name and are categorized are things that we can pursue. But in no way does that begin even to describe all the ways in which you can think about a cell. And in particular, there's many things that just don't follow these strict boundaries. And so, for example, thinking through the lens of gene programs can complement the way of cell types, but it is not yet fully reflected in these integration efforts, although there's a lot of research that goes around that. A second major challenge is balancing between the desire to harmonize things and make them look the same, and the desire to capture the inherent variation that is always part of biology. And that's a variation between conditions, locations, individuals, and so on. And what's important to understand is there's no, and there's never gonna be, one correct answer. It depends on the question that we ask. So if we want to build a simple reference, for example, define all the T cells across individuals, all the T cells in the body across individuals, you want to remove some of the biological variation, for example, between different people. But if you want to study inter-individual variation, for example, you're interested in human genetics, then you want to parse out what's common features that let us first relate all of these cells as T cells and preserve the variation between the individuals. And this problem becomes even more prominent when we consider going across tissues and integrating the ultimate human cell atlas, which is actually a whole body integration. Here we know up front that cells, for example, T cells, are the same and different at the same time. So immune, stroma, even parenchymal cells like epithelial cells, they have common features across tissues, and they also have tissue-specific characteristics. So if you try to categorize everything just to one strict category with a one-to-one matching of a cell to a type, that's no longer going to cut it. And we need a more flexible framework. Oops, sorry. I lost something. There we go. Okay, so all these questions become even, all these questions across all of these topics, kind of that's what looms under or above the word annotation and how we actually do it. How do we balance between specific experts, the community as a whole, what algorithms can do for us automatically, how do we bring all of these things together, and how do we annotate besides discrete cell types? And so in session 2 this morning, and in the first breakout, we're going to be covering many of these questions through presentations. We have a panel, I believe, and we have all sorts of open discussions in the breakouts. Now, after these come the many, many things that this first wave integration effort hasn't yet taken on, either deeply or even at all. So, for example, the challenge of integrating data that represent dynamic and developmental processes is not one we've done yet. We need to, but we haven't exactly done yet as a full integration effort. Buzz is looking at me like this, but we need to do it, it's hard. And then moving from molecular cellular profiles to other levels, especially spatial ones, is another one that we haven't taken on yet. Not as a community, we do this in HCA, but as an integration effort. And so this brings me to the second major theme, which is Atlas expansion. So HCA was never about RNA profiles of cells. So I chose to myself the little tag that says first HCA meeting, not because this is my first HCA meeting, it's not, but because I was at the first HCA meeting. And even then, in 2016, how shall I put it? It wasn't only about single-cell RNA-seq then either. You can go, well, we didn't stream that meeting. We only started streaming 2 meetings down from there. But we have all the materials. People were already talking about all the things we are talking about now, even though we didn't have great tools yet, lab or computational for some of them. So it was always about all the features and properties of human cells. And these additional features are now taking center stage. So yes, single-cell RNA-seq gave us a fantastic first start. We're very grateful to it. But the cellular branch of the atlas, as we called it in the white paper, it includes many other features that are measured at single-cell resolution. I don't know, you have your chromatin organization, your protein levels, your protein modifications, your metabolic profiles, your microscopic images with imaging, dynamic events. We can measure today's transcription rates, translation rates, and then thanks to multiomics, you can also measure all of them simultaneously in one cell. And then on top of that, to understand cells truly, we have to know where they're located, both with respect to some abstract common coordinate system or framework, CCF, and with respect to each other in the tissue. And so this is what we call the spatial branch of the atlas. And now there's been many technical and algorithmic advances, and it's progressing a lot more rapidly. And then finally, on top of all of these, for this to be a human cell atlas, that atlas must reflect human diversity. And that's in terms of ancestry, gender, age, and geography, and many other things as well. And so this first integration effort looks just at one view of cells based on RNA profiles. At the same time, the community is pushing along to collect on all these other dimensions. And as these additional views are growing, we have the opportunity not just to integrate, but to unify. That means to build a unified representation and a unified understanding of what it actually means to be a cell. And that's very different than just saying a cell has these and a cell has that. You want to connect these different views. You want to be able to computationally learn how to predict one view from another. And you ultimately want to understand how one level of organization yields another level of organization in a cell. And so we're going to spend a lot of time in the coming days on this atlas expansion. Session 6, for example, is going to focus on spatial methods. Session 7 is going to look at how adding these new dimensions helps answering new types of questions. And in session 8, we're going to look at how machine learning in particular, to Gary's point about the great opportunities with Mila and Vector, can help us integrate not just across data sets in one modality, like in this first wave, but also across these levels to unify this atlas. Okay. So now as this atlas grows, we should care not just about how we assemble it, but how people use it. And the core question is how we query the atlas to help us answer questions, and that's the third theme of this meeting. And so, as in many other data-rich fields, the scale of our HCA data opened the door for us now to learn and query foundational models of cells across the human body. And we're starting, hopefully this works, and we're starting to see these kinds of models emerging now pretty quickly. And they're relying on different types of machine learning paradigms. You have work with transformer-based large language models. You have work with metric learnings. You have all sorts of things. What matters is that we need to think about how we can harness the tools for what we want to do. And so we're going to spend some time on how we can use these frontiers, these computational frontiers, in order to help us assemble and query a cross-body human cell atlas. And we're going to do this in session 8, focused on foundational models, in session 11, on where the frontiers of machine learning meet the human cell atlas, and in breakout session 12, on building the whole body atlas. And being here in Canada and this partnership with Vector and Mila really helps us bring, we hope, the best minds to think about this problem. Okay. And then finally, this time at this meeting, we're actually pushing beyond the scope and remit of the atlas in the strict sense, and we're going to think on how the atlas, as well as the techniques that our community develops, can help move from descriptive cell annotations to causal understanding of cell function. And so think about it like this. Right now, strictly speaking, from the kind of data that you could collect from, say, single cell RNA-seq of some tissue, and the kinds of tools that we have, you can readily ID cells, you can categorize them, you can label them with all sorts of names or give them a new name if no one has seen them before. You can say where they are at different levels of resolution, and you can describe in which conditions you found them, healthy or disease. All of these are extremely useful things. But unless you already happen to know what a CD8 T cell does, how it recognizes another cell and then how it kills it, our data, that kind of profile, wouldn't actually tell you that that is the cell's function. We happen to know it from before. And it's useful to say this is that, and it has all sorts of clues, but it doesn't give us function. And so at the close of this meeting, we're going to take the cell function question on. And as I said, this is not what the atlas is expected to deliver. We didn't promise this. This is not its goal. But it is something that both the atlas data itself and all of the methods that we're developing enables. And I also think it's something many, many of us care about very deeply. And I'd venture to say that it's probably one of the reasons most of the people in this room actually work on this, is that they want to understand what things actually do and what their function is. And so in session 14 in plenary, we're going to hear several perspectives on how we should think about cell function. And then for those who are interested further, we're going to dig deeper into this in part of the breakout sessions in session 15. Okay. Before I close, I want to spend a couple of minutes on the substantial efforts of all our working groups and committees that keep HCA going. It's a lot of work. We have 4 working groups: analysis, technology, ethics, and equity. Some of them will meet during the general meeting, and in particular, the ethics working group is welcoming all who are interested in this to attend their committee meeting and join them at the lunch break today. I also will highlight the work of our equity working group. I showed you already in the beginning that HCA has come a long way in making sure that we're a true global initiative, and this is thanks for the effort of this working group. That's how we can be diverse in our data and diverse in our scientists, and our scientists can do this work within the community where they live and serve. And this involves a lot of dedicated groundwork, and one of the nice things that happens is these workshops, which we've had this year in Latin America, in Asia, and in Africa, and soon we're going to have one in the Middle East, right? Yes. And so as a great example of those, one great example of those is this recent HCA Africa workshop that was put together in March. And in this meeting, this was a 4-day workshop. It brought together 60 experimental and computational biologists from 11 African countries. I don't know them by heart, but I wrote them down for myself. Ghana, Nigeria, Ethiopia, Zambia, Malawi, Mali, Cameroon, Kenya, Gambia, Senegal, and South Africa. And the participants were evenly split between computational and experimental. They had parallel tracks for computational and experimental activities, a lot of hands-on training, and so when participants left the workshop, they could bring back this work to their own labs and research. Now, all this work in HCA leads to a lot of papers, 128 to be exact, that are either published or in preprint form and already HCA branded by their authors. And that's done with the help of a diligent publication committee. And there's going to be more. And so next up, we have the 3 publication bundles with about 85 papers in them. And they've just started making their way through Nature Publishing Group. Keep your lookout for emails from Ellen. This bundle has 3 tracks, one for analytical methods, one for developmental biology, and one for organ or system-specific atlas integration. If you have any questions about the bundle, but also if you have a paper that's actually ready and you didn't sign up for the bundle and now you changed your mind and you would like to contribute, it's not too late. And you can email Ellen at the address here, or you can find her, I assume she's somewhere here. She is, and she has a very HCA colored shirt to make it easy to find her. Okay. And then, as always, HCA is just an open initiative. We don't have any money of our own that we hand out to people. I'm sure you all know that already. It's only possible because we have a nice partnership with many, many funders who are themselves part of HCA. And they support HCA researchers through many different initiatives and grants and mechanisms. And this is even more augmented by the collaborations that our scientists in many ways have in the context of disease atlases, because you can only understand health in the context of disease and vice versa. And so we're very grateful to all the funder partners. I'm very grateful to all of you for joining. It's so nice to be here. I wish everyone a wonderful general meeting and a fun 3 days. Thank you very much. And now I'm waiting for Gary to tell me what to do.
Thank you very much, Aviv. You were perfectly on time. It's amazing. I didn't have to do anything as a chair to work on that, so thank you very much. Do we do questions? Or
we can do, well, you're the chair.
Yeah.
I believe we can do questions, but sometimes we always run out of time.
A couple of questions before we move on to the next session, if there are any.
Yeah, you need to continue holding your hand up because they have to get you the mic because the virtual participants wouldn't hear you otherwise.
Okay, you want to go first? Okay.
Yeah.
Oh, there's online questions, yeah.
Yeah, we've got an online question from Dana. Gary, do you think the liver is the best atlas?
Dana is clearly in good
Where's my
form.
liver badge? Yes, and we will be giving out liver stickers to people who want.
I want a liver sticker.
And we will be hearing from a really great liver story later as well. One more question here.
Oh yeah, sure. So this is my first HCA meeting. I don't know if I'm out of my spot, but I just want to ask, I asked this in a virtual meeting before, but when you mention all human cells or all human cell types, how many would be those, or at least in an order of magnitude? In the thousands, in the millions, in the billions, are 37 trillion cells on average in a human body. How many cell types is the atlas about?
So I'll take a stab, and I also see that we have Sarah online, and it's one of these foundational questions that's fun to muse about a little bit. So first of all, there's a big difference we now realize between the organs and systems. Probably the brain sits in a category separate from all the others in terms of its very fine resolution cell typing, discrete categories in very large numbers. In many other organs, you would have definitely over 100 or more in something like the gut, but then it gets complicated because of what I said, because of how they're similar and different at the same time. So you look at the small intestine and the large intestine. Just choose a tissue I happen to like. And you can say, well, I have tuft cells in both, or I have an epithelial stem cell in both, but it's kind of colored a little differently. Even within the small intestine, the three major parts of the small intestine, you will see a little bit of a difference in its epithelial cells. So do you count that as different things or as the same thing? Depending on how you do the counting, the numbers will start shifting quite rapidly. And this is why thinking just about the types is too narrow of a definition, and why we wrote, even in the beginning, the types and properties, because types is actually only one way in which you can look at cells. Having said that, I haven't been around during the Human Genome Project, okay? But one of the things you always hear from the old-timers is how bad it turned out for the people who made guesses on how many genes are in the human genome. So I promised myself before this even started that I'm personally never going to venture a guess. We'll just let I think the interesting question is, how much can we predict now, given the data that we have collected, how many more there are to go? And I'll turn to Sarah. And hopefully it works AV-wise.
Sorry, what was the question about cells?
A number of cell types, how many do you think there are?
Not a trick question. Well, we are, when we focus on individual tissues, like, the different tissues of the heart, the different tissues of the intestine. So Aviv has mentioned large, small intestine, different regions of small intestine, and so on. For each of those tissues within an organ, we are in the region of 100. For blood, we're sort of upwards of that. of course, if you take together all of those tissues, in the heart we're talking about 30 tissues, in the brain 100 and so on, and we take the union, we're going to be at a much higher number. And that's really all I'm going to say on that topic for now. I think I'm sure we'll hear more, we'll get more insights throughout the meeting during the days ahead.
Okay, thank you very much, everybody. I'd like to move on. Do we have one more question?
There.
Okay.
Yeah. He was the first to raise his hand.
Okay, sorry, I didn't see that. Go ahead.
I wanted to ask.
Actually, could you introduce yourself? I think that would be great for participants to say.
My name is Nadav. I'm from Sarah's lab. Sarah and John, and so part of the HCA community, but it's my first meeting, so I'm happy to be here. I wanted to ask how, because there is one aspect that I think maybe is not addressed enough, which is kind of like the native state of the cells in the tissue, which we, obviously from obvious reasons, we extract the samples from deceased individuals or after immense processing. So how much do you think can we bridge that gap to and how much of the function are we losing actually by the methods that we're using to probe the cells?
I think it's an awesome question, and it is addressable in multiple ways. There isn't a single answer. I think looking from multiple directions, it's like it allows you to triangulate where reality is, because all of them have some biases and skews. The first thing I'll say is that it really depends on the lab technique that we use. So in certain lab techniques, we introduce more difference from the native state in the tissue when it was obtained than with others. Dissociating cells is likely worse than extracting nuclei. Extracting nuclei is worse than doing it in situ in the tissue. As the methods have advanced, so are we getting closer to what we think is happening there. But the mere act of, excising a piece of tissue from an individual is going to make a difference. So that's one layer, I think, of difference. In some cases, we have the opportunity to obtain tissue that is probably completely healthy. So, for example, since we mentioned the gut, people go through routine colonoscopies after a certain age. Most of them are perfectly healthy. There's no reason for the colonoscopy except for a screening test. Those kinds of samples tend to be very reflective of healthy, quote-unquote, tissue. However, those usually tend to be limited. They won't be as deep, you won't see as much. There's a price you pay for that. Having both looks, having a look from postmortem tissue, from resections in surgery, from what we call adjacent healthy tissue in cancer resections, from biopsies, the more it looks consistent, the more you believe this is the native state. So that's a second whole pot of things. And the third one, which I alluded to a little bit when I talked about the different levels of organization, the more you relate different levels of organization, the more, again, you're closer to the unified actual latent space of what things really are versus the views of them that you see through the lens of measurements and so on. And in particular, if we get there, and I would say there's optimism that one would get there, you would get to the point where you might be able to use live methods and predict from them and generate from them what the molecular profiles are. And if you would be able to do that, that's, further in the future, but, one can dream. As you project like that and you go live, then you would be in a very different place than the one that we are at today, and also you would have better benefits for patients. That's why this multi-scale work is so fascinating and interesting. And in the meantime, we also get a lot of utility from the limited methods that we have today. I think that's a good place to stop.